So I thought it'd be fun if we all went around and said our name and a little something about ourselves. I'll start. My name is Rod, and I like to party.
My name is Jake, and I like biotech.
My name's Lila, and I like elegans, which is funny because we were working with elegans to shut off a specific gene by using RNAi to silence the gene.
In order to do this, we chunked the worms from a starter plate, which is a petri dish with wild-type worms on it, to a feeder plate, which is a petri dish with the RNAi feeding strain we are assigned, which turned out to be DPI-11. On this feeder plate, the worms eat the bacteria that silences a specific gene. In this video, we are looking through a microscope at a starter plate with the wild-type C. elegans. Most C. elegans are hermaphrodites, and there is a very small population of males. Here we see eggs, L4s, adults, and some L2s in the top left corner. The C. elegans lifestyle consists of 6 stages: the egg, the L1, the L2, the L3, the L4, and the adult.
When the camera moves, you can see all 6 stages of life: the adults, the L1s, the L2s, the L3s, the L4s, and the eggs. You notice that the worms are very transparent and you can see through them. The adults have eggs inside, which are the dark area around their stomach. The media that they swim in has all of the bacteria that they eat, and the worms, as you can tell, move very easily and smoothly through the media because they're so long and skinny.
In this video, you can see that almost all of the C. elegans are now much shorter and fatter, about 80% knockdown. This is what we expected from this mutation because the feeder plate we chunked the wild-type worms onto contained the DPY-11 gene, which inhibited their ability to grow. You can see that this is troublesome for the worms because it is now much harder for them to move throughout the media. It looks like the DPY-11 gene shows up when the worms are between the L1 and L2 stages of their life cycle.
We concluded that the dumpy gene had to do with the length of the worm. The dsRNA entered the cell and triggered the RNAi pathway. Dicer then chopped up any dsRNA it found into short pieces of RNA called small interfering RNA, or siRNA. The siRNA then bound to several proteins, forming a complex called RISC, or RNA-induced silencing complex. The double-stranded siRNA are unzipped, leaving a single strand of RNA. RISC probes the cell for mRNA that it recognizes. RISC binds to the matching mRNA and knocks it down. The mRNA can no longer be translated into a protein, therefore the gene has effectively been turned down or silenced.
I thought this lab was really cool and interesting because of all the real-life applications of silencing genes with RNAi and how it has helped cure diseases and will most likely lead to curing more diseases in the future.
I really enjoyed the lab. I thought that the worms were fun and interesting and cute, especially the dumpies. Thank you so much for watching.